Hello, everyone, and welcome to today's webinar, Investigation of Endothelial Cell Viability and Growth on 3D Printed GelMa Vascular Networks, presented by Chrisoula Chappell, postgraduate student, University of Manchester. I'm Christie of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com/Cell Culture Heroes. Now let's get started. You may submit questions at any time you want during the presentation. Just simply type them into the Ask a Question box and click Send. Your questions will be answered via the contact information you provided at the time of registration. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of your presentation window, or report your problem by clicking on the Ask a Question box located at the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of your presentation window and follow the process to obtain your credits. I'd now like to introduce today's speaker, Chrisoula Chappell, postgraduate student, University of Manchester. For a complete biography on Chrisoula, please visit the Biography tab at the top of your screen. Chrisoula, you may now begin your presentation.
So, hi everybody, my name is Chrisoula Chappell. I'm from the University of Manchester. We're predominantly the Biomaterials group, and we're based at the North Campus, really. We're the cast-off, but we're good people. So I'll be talking to you about the investigation of endothelial cell viability and growth on 3D printed GelMa vascular networks. Now, GelMa is for gelatin methacrylate, a hydrogel. So my supervisors are Olga Suku and Professor Brian Darby, that are very well known around those fields. So if we just go into slide 1 to give you a little bit introduction. So the reason why we're doing this project is because about breast cancer. These are UK statistics in the NHS. So in 2017 alone, 63,410 women were diagnosed with invasive breast cancer. Now, invasive breast cancer means it's spread outside the lobes of the breast. Now, the most predominant one is DCIS, which is ductal carcinoma in situ, and it's a very common form. And as you can see from the histology, which has got the pink and the purple stain, it's when the breast ducts of where the women produce the milk actually get cluttered with malignant cells, and that's the most invasive form of breast cancer. Now, women, because when they inherit that disease, they tend to do breast augmentation, and that involves complete augmentation. They lose their breast completely, and that can hamper, their confidence. So what surgeons have tried to do is they have tried to put fat tissue up there to restore the breast, but because there's no vascularization, obviously the breast tissue dies. And as a result, a lot of scientists now are going into the tissue engineering mode, and we're trying to figure out a way to introduce artificial vascularization for these women. So if I go to my next slide, what makes a successful 3D tissue development, and does it really need vascularization? The answer could be yes. The artificial vascular networks are first, either printed or they're casted, but we're going to use the 3D printing method. We use a simple robotic printer, and it prints out a Pluronic F127, and that's a little bit also of a surfactant, and it's a sacrificial material. Now, a lot of people have used Pluronic F127 because it's easy to be rid of, which is the opposite to wax or sugar. Then gelatin methacrylate, our actual hydrogel, is casted on top, and what we want to do is we want to put endothelial cells within those hollow channels. The adipose-derived stem cells, which is basically fat tissue, are basically encapsulated within the hydrogel, and what you want to do is you want to create a feeding system for both the helix and the adipose, and you want them to co-culture together and basically do what the surgeons do, but you want to create viable vasculature, but it's artificial. Now, as you can see from my little picture over here, these are artificial channels that we printed here at Manchester, and we actually put a fluorescent dye through. Now, the fluorescent dye shows that there's no obstructions and that there's barely any air bubbles, and that our structure has worked. It hasn't collapsed. And it also basically shows that, with these, we can actually put cells through our structure, which is good news for us. And the reason why we've done a bit of a hexagon is because we want to make it look more like vascularization, so forth. People ask me, well, how do you do these channel formations? Well, we use the robotic printer. The robotic printer works like a simple bioplotter. So it actually prints Pluronic filaments on glass slides. And the way we actually want it to actually be successful is we want to functionalize those glass slides with ammonia, hydrogen peroxide, and a saline. Now, as you can see from the little diagram that I've basically created, your glass slide is with the ammonia and the hydrogen peroxide, it's cleaned thoroughly of any kind of residue. So it's absolutely crystal clear. And then you put your silane on top, because your silane is the one that's going to attach to the gelatin. And you basically make sure that, when you do cast your gelatin on top, it's not going to detach in the DMEM, which is the media conditions at the cells, and it's proven to do that. So once you do your glass slide functionalization, we use the robot printer, and the robot printer can do a variety of shapes. It's got over 92 programs. We tend to use roughly, I would say, about 12 programs so far. We draw circles, we draw a lot of polygons, we draw so many things at the moment. And what we have achieved is we have achieved a 300 micron diameter in each channel. And as you can see, we have created those little channels and on the glass slide. And then Pluronic obviously has a lower critical temperature, so it has to be printed at 5 degrees or so, because obviously it solidifies at room temperature. Well, 17 degrees, I would say, or above. And then you would cast your gelatin methacrylate on top, really. And then what you do, well, gelatin methacrylate, because you have added the methacrylate group, it needs to UV polymerize to become a proper hydrogel. And we put it in a UV lamp. It's an LED lamp. We actually use something that people use for their nails. It's very, very low spec, but it does the job. We leave it there for a couple of seconds. Once the gel has basically formed, what we do is we prick it at the end where the channels are, and we drain the Pluronic after we've put it on ice, because obviously with the ice the Pluronic becomes a liquid again, and you flush everything with water and you're left with a hollow structure as shown in the previous slide. So you're going to say to me, well, why are you doing this? The reason why we're doing this is because we want the HUVECs, which are the endothelial cells, to go in the slides, invade those channels, and create the artificial vasculature. So you're going to say to me, okay, well, what are your general project aims then in that case? So here's a slide for these people in general. So our project aims, well, my project aims predominantly, it's me doing the work. It's basically doing a 3D viable vascular tissue construct, okay? And we encourage the stem cell differentiation within the gelatin methacrylate. And what we want to do is we want to create those 3D channels with the 300 micron dimensions using that simple bioplotter. Then we want to cast our hydrogel on top, and we want to basically make sure that the HUVECs and the adipose-derived stem cells can grow over time. And what we want to do is we want to create a viable kind of artificial tissue for people that need it. So we're going to say, so how is gelatin methacrylate made? It's a common biomaterial, I would say. A lot of people have done a lot of studies on it. It's made by different methods, really, but the most important method, and a lot of people are using, it's known as the one-pot synthesis. Now, the one-pot synthesis is a very ideal method because, it doesn't use large amounts of methacrylic anhydride like the conventional method, and it doesn't need multiple pH adjustments like the sequential method. So what you do is I've created a little diagram for you, as you can see, and I've created the chemical bonds for you to follow. What you do is you put your buffer up there with your gelatin. So, and then what you do is you submerge it in water, and then you stir it until it becomes crystal clear. Now, once it becomes crystal clear, you start start doing your pH adjustment first, because you want a pH of 9. Then you go into doing by adding methacrylate anhydride, and that's drop by drop, so you can get a full dispersion. And then you leave it for 3 hours for the reaction to occur. Once that is done, you do a further pH adjustment so that it could be near to 7. And then what you do is you put it into a little pocket, and it's a dialysis tubing. And basically what you want to do is get rid of any excess methacrylate anhydride, and you leave it for a week. And that's how you get your really nice pure product. Now, the things that I haven't included in that diagram afterwards is that basically you have to freeze dry it. And the reason why you want to freeze dry is you want to get the water out as much as possible by forming very small crystals in it, and then you get a foam. And once you get this foam, you add either PBS, DMEM, or water, and then depending on the calculations and the media and the gelatin methacrylate percentage you want, that's the amount of the solutions that you add. So then you're going to ask me, well, how do you know that the methacrylate anhydride has attached? So that's when the next slide comes in, and this is through NMR analysis. Now, this is known as Proton NMR. So it's liquid NMR, and basically what you want to see, what we did is we wanted to correspond our findings with the graph from a paper, and that's a particular very famous paper from Shirahama et al., which actually did the one-pot synthesis, and we optimized it quite a bit. And what the most important thing is that you should actually view this, the green box, because the shorter the peak there, it means the less free lysine groups, and that's where the actual methacrylate anhydride attaches and so forth. And that's the most important peak. So if you've got a very small peak, then it means you've got a lot of methacrylate anhydride. There are certain assays that you could do potentially to see, how much you have got attachment, like TNSB assays. But in general, people tend to use the NMR method and so forth. If you want to, people have also discussed really if they want to look at it further by doing a further hydrolysis experiment. But however, that's very time consuming, and also because of the complications of gelatin and the way it's denatured from collagen, that's very, very hard to predict because it can be cleaved at different methods. So people just use the standard NMR. So then I decided with my group, well, what happens after, we photopolymerize it? Can we see any additional peaks? Can we see any more chemical bonds? And we can use the liquid NMR. So we decided maybe we can use the carbon, which is a solid NMR. Unfortunately, because it was too wide, the plane, we couldn't really distinguish certain bonds. But what we could see was basically, we could see a whole handprint of the gelatin, really. And basically, the peaks that are basically the very, after the big peak, the ones that have got the widest part, really, they're all the different groups within the gelatin. And that's the most important. People do actually use this method. I know Aliev et al. Did, to distinguish whether it has been, hydrolyzed appropriately from collagen. And that's what we got, really. It's a fingerprint of gelatin methacrylate. So although we didn't get our results, we didn't let our spirits go. We want to see, well, how hydrophilic is our gelatin, or how hydrophobic it is. So we did some contact angle studies, and it turns out that gelatin methacrylate is very hydrophilic. It's 21.2. So if you've got a value that is less than 50 degrees, that's a very hydrophilic hydrogel, which is very good. It's ideal because they'll love being in a hydrophilic environment. So because we're printing with Pluronic, and obviously there will be, maybe we can't guarantee the whole of the Pluronic leave-in, saying, well, how hydrophobic is Pluronic and so forth? And it actually is very hydrophobic. It's around 75, which is basically to the more than above threshold, really. But no, other than that, though, we decided to carry on with our cell studies. So we did cell studies and we did encapsulation versus a 2D cell culture. So we used the adipose-derived stem cells, first of all, and what we did is we grew the adipose-derived stem cells, and we did different time points, day 1, day 4, day 7. So within the encapsulation, as you can see, this adipose-derived stem cells from day 1 actually elongate, and they're trying to invade the gel. But if you do a 2D cell culture on them, they actually form little clusters. And I should point out that we did light microscopy, and then the ones next to it are the live dead. So the green stain predominantly is calcine, and that shows that the cells are alive. And then the third one is basically when the cells are, we're looking at the cytoskeleton, the actin of the cells, and we're looking at the nuclei as well. So the more organized the actin is, the more likely the cells are very healthy and they're willing to proliferate on the gel, which is good news for us from day 1. Then we go on to day 4, and as you can see, the cells have actually exploded within the gel when they're encapsulated. Not only have we not found any red dead cells, but the actin is so well organized and it's spreading out, but that's really encouraging. But we've also seen that phenomenon with our 2D cell culture, which proves actually that the cells, whether they're encapsulated or whether they're on top, they still like the environment. Then you go into day 7, and the cells are still looking happy, they're still proliferating, and there's barely any dead cells. In fact, there's none really. And what you want, and these are positive results, and that's what you want to see within your cell line. So basically, our adipose-derived stem cells are more than happy to be encapsulated up for 7 days, and we're assuming that they would be happily encapsulated even after the 7 days. So if we go into the next slide, we did a control, and basically this control group is the human dermal fibroblasts. Now, human dermal fibroblasts are found in your skin. They're very easy to grow cells. They're not stem cells, they're a primary cell line, but however, they were used as our control. So for our encapsulation, which is basically around this box, as you can see, we did it the same as the adipose-derived stem cells from day 1, day 4, and day 7. So day 1, again, you can see that the cells are actually trying to spread within the gel. They're really elongated, they're well organized. And on day 1 on the 2D cell culture, they're forming clumps still, but they will get there. And as you can see on day 4, they started to form different cells from the clumps on top. And then within the encapsulation as well, they're starting to spread more. And by day 7, really, they've invaded predominantly most of the gel. And again, there's barely any cell death. And, the actin is very well organized, which is something that we really want to see. It's very positive results. Then we go on to our human umbilical vein endothelial cells. Now, the human umbilical vein endothelial cells, we only did a 2D cell culture study. And the reason for that is because the human umbilical vein endothelial cells are not going to be encapsulated within our gel. We don't want to do that. We don't want to create necessarily that artificial environment for them because they are sensitive cells. From the name as well, they are derived from the umbilical cord of babies. Now, the babies are born, and then the mother or the parents can donate their cells freely. And what you get really, so we used a pooled version. So all these cells are actually from different donors, because you get to see a variation really. You don't want to constrict it genetically to one individual. And what you can see, the human umbilical vein endothelial cells are a bit more sensitive. They require up to day 14 to fully expand within the hydrogel. That's not because there's something wrong with the hydrogel, it's because of the way they grow, really. So on day 1, as you can see, they are forming clusters like the other 2 cells did. And then on that was day 1, day 4, as you can see, more cells can be visible. Day 7, they're forming colonies, and then by day 14, they've expanded into the hydrogel, which is positive, and that's what we want to see. Eventually, when you actually want to create the artificial vasculature for them, the UVEX will eventually invade that hydrogel, and what we want to see is we are thinking of potentially adding an artificial pump, and we want to see whether there be any differences to, the way they actually behave as to having a static culture, really. So if we go further on, so have we done any kind of preliminary studies? So I'm going to conclude with that. Yes, we have printed the channels, and preliminary results have shown that we have put HUVICs in there. Now, we've only done it for day 1 till 2, really, because it was only a little test that we wanted to do. And it has shown that the cells do attach, actually, and they do survive. But we need to actually encourage that further. Now, we've noticed that they do want to elongate as well, but because we only went up to the 48-hour point, it would be interesting to see what it would be like after 14 days. However, we did notice that even though you did get attachment on the gelatin parts of the hydrogel, because obviously they have got the glass slide underneath, there was hardly any attachment there. So we're thinking maybe if we add some gelatin or fibronectin or something, some cells can grow on that part as well. And the other thing is we've noticed is that even if we've got 2D or an encapsulation experiment within our cell culture, we've noticed a high cell viability. And that's the most important thing, is that our cells enjoy the environment and they're not constantly dying. They're happy to divide, and they're not even differentiated at that point, really. So we don't know. We might go down that route. I should point out that we also did more confocal studies as well with nanoparticles. These are fluorescent nanoparticles, as you can see from the fluorescent image on top. And basically, we wanted to make sure by putting fluorescent nanoparticles, there was no obstructions, there were no leaks in our artificial channels. And we've proved that, which is positive. So I would also like to conclude, well, what is your future work going to be like? I mean, you've got a lot of work ahead of you, but what do you actually want to achieve at the end? We predominantly want to achieve basically expanding our 3D printing and basically co-culturing our HUVICs and our adipose-derived stem cells together. So by culturing our HUVICs and seeing whether they can survive after 48 hours within that environment, then additionally adding the adipose-derived stem cells, it'd be really interesting to see whether we get any markers like CD31 or VE adhering and EE adhering, which forms, when blood vessels form. So also we want to potentially start introducing an in vivo bioreactor, because then you are creating an artificial tissue, an artificial environment, and you want to see them proliferating. We actually, I've included some hydrogel characterization because we want to know more of our hydrogel. We are currently doing that. We've done a lot of surface tension and rheological analysis and DMA analysis, and we found some interesting results there. But at the moment, we're mostly interested about how our cells are going to react, because this is the biggest thing, really. And if we can make this a possibility, then, we can advance the project furthermore than we have thought. And, it would be incredible for our work, really. So I would like to thank you for listening to me. I would like to thank Professor Brian Duffy, Dr. Olga Tiku, Faye Zeng. Faye worked really hard with me, bless them. He worked countless hours with me to get this far. So big thank you to him. The EPSRC, it's our funding body for the engineers. And I would like also to thank the people that have given me the opportunity to do this presentation. And please feel free to let me know if you do have any questions. So any questions, please do let me know. I'll be more than happy to answer. Thank you.
Thank you, Chrisoula, for that informative presentation. And as a reminder, we are taking questions, and those questions submitted now and those submitted during the on-demand period will be answered via email to the contact information you provided at the time of registration. Now, I'd like to thank Chrisoula for her time today and for her important research. And I would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us. And don't forget to check your inbox if you did submit any questions. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. You will now be redirected to our upcoming Cell Culture Heroes webinar on November 21st at 9 a.m. Pacific Time. The webinar is titled The Role of Immune Cells and Immune Checkpoints in Multiple Myeloma, presented by Natalie Honisch, PhD candidate, University Hospital, Bonn, Germany. We hope to see you there.